Hello everyone, the topic is all about photosynthesis. Know what is photosynthesis? A photosynthesis is a chemical reaction which converts the light energy into a chemical energy. And it is also an anabolic process in which the smaller molecules are converted into complex form of molecules. Photosynthesis is a redox reaction in which the both oxidation and reduction reaction take place simultaneously. In oxidation reaction, the water is oxidized into oxygen, whereas in reduction reaction, the carbon dioxide is reduced into carbohydrates. So the given reaction is the photosynthesis reaction. Photosynthesis reaction occurs in the chloroplast of plants and it is a double wall membrane bounded organelle. It is usually 4 to 10 micrometer in diameter and 1 to 33 micrometer in thickness. The stroma is present inside the chloroplast and within it a thylakoid is present inside the stroma. So here pigment system 1 which is present in the outer thylakoid membrane and pigment system 2 which is present on the inner membrane. The chloroplast contains proteins, phospholipids, chlorophyll, carotenoids and ribosome and circular DNA. Photosynthetic pigment searches, chlorophyll, carotenoids and phycobilins are present inside the chloroplast which is responsible for photosynthetic reaction. The chlorophyll searches A, B, C and D and bacteriochlorophyll like A and B. Chlorobium chlorophyll 650 and 666 are present. Carotenoid searches carotene and xanthophyll and phycobilin searches phycoxanthophin and phycoerythrin are present in the chloroplast. Photosynthetic unit known as quantosome. It is located in the inner membrane of thylakoid lamellae. Usually measures about 180 angstrom into 160 angstrom. So these two scientists Steinman observed granular structures in the chloroplast, lamellae and electron microscope. Later, Park and Begnins confirmed these granular structures as a physiological unit of photosynthesis and coined the term quantosome. Absorption spectrum is defined as the amount of absorption of different wavelength of light by the pigment. And the action spectrum is defined as a curve shows the rate of photosynthesis at different wavelength of light. The light reaction. It is a photochemical reaction which takes place in the thylakoid membrane of the chloroplast. So it has two phases, photooxidation and photochemical phase. In photooxidation phase, first the light is absorbed and it is transferred as an energy from the accessory pigments and it is carried out to reaction center. So there the chlorophyll A is activated. And then photochemical phase where the water photolysis takes place and oxygen is evolved and then the electron transport chain enters into it and energy molecules are synthesized. System and reaction center. So the thylakoid membrane consists of photosystem 1 and photosystem 2. Photosystem 1 is an unstacked region of granum facing stroma of the chloroplast, whereas photosystem 2 is stacked region of thylakoid membrane facing in the thylakoid. Photosystem 1 consists of core complex 1 of reaction center P700, light harvesting complex 1. Photosystem 2 contains core complex 2 consists of reaction center and P680 and light harvesting complex 2. And then photochemical phase. In this the electron passes through the electron and then the power such as ATP and NADPH are synthesized. And then photophosphorylation. Cyclic photophosphorylation. Here photosystem 1 is involved and only ATP is synthesized. No NADPH and oxidation is evolved. And non-cyclic photophosphorylation, both photosystem 1 and photosystem 2 are involved. Here ATP and NADPH are utilized and oxygen are evolved. Dark reaction is also known as biosynthetic phase. It occurs in the Stromo of chloroplast, ATP and NADPH is utilized from the light reaction, and carbon dioxide it is reduced to carbohydrates. For dark reaction, light is not required, and it is also known as Calvin-Benson cycle. The dark reaction consists of three phases. The first one is carboxylation, and then reduction, and finally regeneration. Step of carboxylation. The enzyme ribulose carboxylase oxygenase involved in the formation of 6-carbon compound, which is more unstable, and this 6-carbon compound is broken down into two molecules, that is 3-carbon compound of phosphoglyceric acid. In the next step, reduction, the formed phosphoglyceric acid is phosphorylated by the presence of ATP, and it produces 1,3 bis phosphoglyceric acid by the enzyme phosphoglyceric acid kinase. And then the 1,3 bis phosphoglyceric acid is again reduced to glyceraldehyde 3-phosphate by using the power NADPH. Finally, triose phosphate is formed. Step regeneration. The ribulose biphosphate involves various intermediate compound formation. And finally, the one molecule of glucose is formed from the 6th molecule of carbon dioxide. Hatch and Slack pathway or C4 cycle. So this reaction involves two phases. One is mesophyll cells and then bundle sheath cell. The first phase takes place in the stroma of mesophyll cells. 3-carbon compound phosphoenol pyruvate is formed to 4-carbon oxaloacetate. Oxaloacetate is a 4-carbon compound, so it is also named as C4 cycle. The oxaloacetate is formed and then it is transported to bundle sheath cells. So in the bundle sheath cells, malic acid, that is oxaloacetate, undergoes decarboxylation reaction. Then it produces 3-carbon compound, that is pyruvic acid, with the presence of carbon dioxide. And the released carbon dioxide combines with ribulose biphosphate and it enters into the Calvin cycle. And then sugar molecule is released into the phloem. The pyruvate is transported into the mesophyll cells. That affects the photosynthesis reaction are quality of light and quantity of light, intensity of light, oxygen and carbon dioxide, as well as temperature. Thank you.